welcome to Generative Energy Podcast. I'm Danny Roddy, and with me is a very special guest from pranarupa.wordpress.com, Phil. Phil, how you doing, buddy?
Good, thanks, Danny. You?
Very good. I can't complain. And I'm extremely excited about today's topic, which is the cell and the protoplasm versus membrane pump. So, I mean, the first question I think that's relevant is like, what is this topic and why is it important?
I guess this topic is concerned with how energy animates life. Is it through some kind of mechanical membrane-bound ion and proton pumps that are kind of studded in fatty bilayer membranes, and this machinery has just managed to jumble itself together through random chance events? Or is biology actually kind of an emergent system that's evolved out of ever more complex arrangements of like fundamental energetic properties of simpler geochemical systems?
Would you say that like the current model of the cell, which is like the membrane pump, is kind of divorced from this like energetic view of physiology?
Yeah, I think it's very much divorced from life. I think it's honestly insane.
So before we get too far, so it's insane. It's not, doesn't follow kind of a logical flow of life. When did you like kind of first get interested in cell physiology, and when did your kind of ideas about it begin to change or drift away from the norm?
I don't know. I've been practicing yoga now for like around 10 years, and early on I was just interested in an explanation of things, how these techniques worked, why they worked, and I started looking into just trying to like more kind of bioenergetic models, and read books like James Oshman's books on energy medicine, and then a yoga teacher mentioned Gerald Pollack's book Cells, Gels, and the Engines of Life, and I read that. And at the time, I don't think it really hit me of exactly kind of how significant the kind of view of biology that he was presenting was, until many years later, I stumbled across Ray's work, and it kind of then it hit me of just how significant that kind of view of biology was.
We got introduced to the work exactly the opposite way. So I started with Ray's work and then got into Gerald Pollack's work. But I think Pollack, that book, if anybody's interested in this topic, Cells, Gels, and the Engines of Life, is just, I think, pretty phenomenal. I read through it every once in a while, and I pick up new things every single time I read it. And the illustrations are especially useful because the complex is so tough to kind of visualize, and I think whoever did the illustrations did a pretty good job.
Yeah, it's a good book. It's definitely very accessible, and you don't need to be an academic to understand it.
Understanding how the cell works, obviously that's important because cells form tissues, tissues form organs, and organs form us. So do you think it's an unreasonable starting place if we're looking at the health of the cell can translate over to the health of the person, right?
Yeah, I don't think you've got any hope of really understanding biology and developing kind of reasonable medical theories if your fundamental ideas about how cells function, how they metabolize, they're completely wrong. And I'm pretty sure that the membrane theory, at least the idea that it's a phospholipid bilayer with sodium and potassium channels and all the other receptor molecules that apparently are studied into it, I don't think that's a very likely model.
The membrane pump theory of cellular physiology serves as the foundation of research on diseases like cancer, AIDS, and other deadly diseases. So, I mean, if we don't know much about the cell, it serves to say that we don't know much about health or disease and sickness in general. So why don't we go over the membrane pump hypothesis, Phil?
Okay, so basic idea is the cell is a bag of free water with ions and dissolved enzymes randomly floating around, contained in the lipid bilayer membrane. And it's a little more nuanced now. There's, maybe some acceptance of limited water structuring occurring. But basically, that's it. This is where the model has come from, an idea that to separate the watery inside of the cell from the watery outside, you needed a membrane.
In addition to that, it's also kind of bound up with this lock and key style receptors, and that's how cells kind of communicate with each other.
Yeah, this very strange kind of clunky mechanical biology. Maybe that's something to get to later after we've kind of discussed Ling's work. But I don't see how kind of the current receptor-based model can explain like the functions of things like progesterone and estrogen, and their kind of global functioning on the cell.
Okay, so the cell is a bag of salty water with proteins dissolved inside and enzymes. Let's go over the origins of the lipid membrane, which was first thought to be impermeable and later found to be semi-permeable. So this presents a huge problem just in itself.
Yeah, I think it was known that it was permeable to water, so it was suggested the membrane was a mosaic. And then the potassium ion was also found to be permeable. And so a potassium channel was proposed, I think, by Boyle and Conway. And channels allow ions to move across the membrane down the concentration gradients. And the potassium channel was claimed to be of a critical size that it would allow the movement of potassium and its shell of associated water, but it would exclude sodium. However, later it was found using radioactive traces that sodium and other substances believed impermeable were in fact permeable. He then had to engineer pumps into the various channels which were proposed for sodium and the other ions, because if you've just got for the various channels, all ions will surely eventually attain kind of equal intracellular and extracellular distributions.
So to quickly review, we have a cell, we have a higher concentration of potassium inside the cell and a lower concentration of sodium inside the cell, and a higher concentration of sodium outside the cell and a lower concentration of potassium. And these are ion gradients, and we have to explain why these ion gradients are maintained. Membrane is semi-permeable, and these ions can diffuse through it, but how do they diffuse through it? So channels were invented for potassium to fuse through it, and the channels were postulated that they were small enough for the smaller potassium ion, but they excluded the larger sodium ion out. And then later it was found that sodium was also diffused through the membrane, so they had to think of pumps. And the channels don't require energy, right? They're just kind of passive. Supposedly.
The channels were thought to be pretty much passive, but I think now the kind of channels and pumps have been kind of assimilated into one thing, the sodium-potassium ATPase, at least in the sodium-potassium pump.
Okay, and then one of the key features of the sodium pump is that it constantly requires energy.
Yeah, according to some estimates, it would require like 15 to 30 times the cell's available energy resources. Yeah, I think the fact that it has had all these various different ad hoc modifications kind of that it's been built from them should have called someone to question, well, maybe something's wrong with the fundamental premise. Maybe this isn't maintained by a membrane. Maybe, sodium, there's some other mechanism involved in controlling sodium and potassium distribution. But apparently no one's thought to do that outside of a few heretics. And they've been basically ignored.
So those people, was Troschen the first?
Yeah, I think Troschen was the first kind of in relatively kind of modern era. But I think there were people talking about the cell as kind of more of a protoplasm, and describing a gel-like substance on the inside. I think Félix Tejada, in the 1800s talked about the cell substance as sarcode, basically protoplasm, this kind of jelly-like substance. So people had obviously noted that it wasn't like a bag of, just a bag of water. There was some kind of something kind of substantial there. And if there's some jelliness there to it, then why was there ever a need to propose a membrane?
Why don't you talk about some of the experiments where they pierced supposed membrane?
There are a number of experiments where they basically done things which should have damaged the periphery of the cell, the supposed lipid bilayer, in a number of ways. These include piercing with microelectrodes, micropipettes, and electroporation. But I think the electroporation might be a different issue. But the microelectrodes and micropipettes and things like patch clamp experiments all will be disrupting the periphery. Like the patch clamps will be removing like a chunk of like the outer skin of the cell, leaving a hole roughly 1 million times the size of a potassium ion. And yet the cell survives and ion positioning is undisturbed.
And if these things were punctured, basically the water and the ion gradient should completely collapse. And I think Pollack says like water out of a dam, right?
Yeah, but it remains undisturbed. And this shouldn't be all that surprising. I mean, there's a genus of algal seaweeds. I think it's pronounced calopa, but it's an organism composed of it's a single multicellular, multinucleated, single-celled organism, which is up to 3 meters long. And this structure can be damaged by fish eating sections of it, and it will continue to function without leaking ions. And it's clearly a gel. And this organism and others like it should call into question the entire reasoning used to propose a membrane. Epithelial cells can be cut into sections. Sections without nuclei will survive for a few days, and those sections with nuclei can survive indefinitely and reproduce. So clearly a membrane can't be that important for cell function. Like the lipid content of cells also varies widely. In humans, I think red blood cells contain around at the very high end, 47% kind of fats, and this is dry weight, and that should be enough just to form a continuous lipid bilayer membrane. But there are other cells like erythrocytes in some birds where the lipid content apparently ranges as low as 4%, and that really doesn't seem like it's enough lipid to form a continuous membrane, even one studded with channels, pumps, and receptors. And again, that's 4% as dry weight with water removed. And if you factor in the water, a lipid membrane seems fairly improbable. A point used sometimes to say, well, look, there must be a lipid membrane is we've got these electron microscope images which show a bilayer pattern. If you can treat cells in mitochondria with solvents to remove up to around 95% of their fat content, stain them with osmium, and they will still produce a characteristic two-dimensional bilayer pattern. And I think that supports Harold Hillman's criticisms of a lot of the imaging procedures used in biology. And his basic point is that the lipid membrane seen in these electron microscope images is basically an artifact of the staining procedure, as when a cell or tissue is imaged by electron microscopy, it goes through a number of procedures. It's cut, fixed in glutaraldehyde, washed, fixed again in osmium tetroxide, washed, dehydrated in ethanol, reducing its size significantly. Propylene oxide is then used to remove residual ethanol.
Sounds like my normal bath.
Yeah, this is basic. And there's more. It's embedded in a mold, and then it's cooked for 24 hours at 60 degrees Celsius, and then subjected to electron bombardment. I don't know. This seems like it might not be producing an accurate image of the cell.
And there's something I had never thought about, but I first read about it on your site. But it's not unreasonable to ask where are the photos of the channels, pumps, and receptors too, right?
Yeah, I think the lipid membrane is only is supposed to be around 10 nanometers in width. So, and I think the receptors, channels, pumps, they should be around 20 to 30 nanometers. So there should be some evidence for them in the electron microscope images, but I don't think there is any.
Enter Gilbert Ling and his famous experiment to see if the cell had enough energy for all of this pumping. And that was what, in 1948?
Yeah. He published his PhD thesis on the effects of metabolism, temperature, and other factors on the membrane potential of frog muscle fibers. But he started to doubt whether the sodium pump hypothesis was valid and the entire membrane model. And of course, that would also mean questioning his own PhD thesis. And I think he did this fairly publicly. He was supposed to give, I think, a talk on the sodium pump. And I think he basically got up there and said, look, there doesn't really seem to be much evidence that this actually exists. And then that he basically said, then stepped down. And I think his professors told him that he should be very careful about questioning the sacred cow of the sodium pump.
But I'm totally blanking. What was his discovery of some kind of procedure?
I think it was the Ling Gerard microelectrode.
Yeah, so that was a big deal, and then after this, supposedly his credibility in the science scene really went out the windows.
Yeah, he just kind of dropped off the map. Despite being warned not to question the sacred cow of the sodium pump, he went ahead and did so and exposed frog muscles and nerves to various agents to suppress respiration and glycolysis. Of course, this should prevent the pumping of ions into and out of the cells, but found that basically ion concentration of sodium, potassium remained fairly standard. They didn't seem to change significantly from control pairs.
I guess I have actually a question about this, but he didn't kill the cell, right? It's just like he brought it to the edge of death?
I think so, yeah.
Okay.
Yeah, I think this suggests that whatever is controlling sodium and potassium in the cell is a fairly passive process, at least when compared to the idea of membrane pumps. And I think evidence of this can be seen in hair, which is composed of dead cells. But if you wash it of all sodium and potassium and then dip it in a solution of sodium and potassium, it'll take up far more potassium than it does sodium. And it seems somewhat improbable that dead hair has, got little sodium and potassium functioning in it.
That seems like a reasonable stance.
But yeah, it's not just sodium and potassium. You also need pumps for magnesium, sugars, amino acids, and even for some toxins. Apparently, there are aminopterin and curarine pumps, and also tetracycline pumps in some bacteria.
These pumps take up space on the membrane, right?
Yeah.
And then how does it pump like a substance it's never seen, right?
I have no idea how it accomplishes this.
Okay, so it seems a little wonky.
Yeah, I think that's an understatement. I think it basically developed as a misunderstanding about the nature of water, and this idea that water was just some kind of lifeless, amorphous substance. I think to kind of understand what's actually going on in the cell, considering the ubiquity of water, you've got to kind of factor in how water behaves. I think this is what Ling's hypothesis does. One of the main features of it is macromolecule water interaction. And this isn't like a new idea. I mean, it sounds like it sounds kind of a bit flaky when you start talking about water structuring.
I like the first time I had heard about this was those people selling like structured water in like bottles, and I think just that poisoned my mind to the idea. And I remember reading about it and being like, oh, this is silly. And then what Pollack goes over, both poly water and memory water, so two giant kind of debacles in science that the people that proposed both of them ended up looking pretty foolish and kind of poisoned the well of water research, no pun intended.
Yeah, I don't know if the polywater thing is entirely. I think there might be something to it.
Oh, okay.
I don't know. Haven't looked into it enough to really say. I think that's changing slowly. I think it's basically been accepted that there is, that water structuring occurs, but it's, it occurs to a fairly limited extent, and it's still pretty much ignored as a biological phenomena.
And we're going to talk about Ling's Associate-Induction hypothesis, but before Ling, there were a lot of people that talked about this very thing, like Albert Szent-Györgyi.
Yeah. He was talking about very, very similar stuff. Also in Russia, people like Troshin and others, Nasanov and Fischer. It was a matter of how water structuring in the protoplasm kind of changed that water's affinity for various ions.
When you say protoplasm, is that interchangeable with the term like gel, basically?
Yeah, pretty much.
And then does protoplasm replace cytoplasm? Or is that just a different, like referring to different parts of the cell?
Protoplasm and cytoplasm, I think, are fairly interchangeable, but cytoplasm is more used more typically.
In the membrane pump?
Yeah, in the membrane pump theory.
So Szent-Györgyi and Gilbert Ling both kind of shared this macromolecule water interaction, and that is making up the protoplasm of the cell. Do you mind kind of expanding on that and like the structuring of water and maybe giving some examples, Phil?
Okay, so yeah, some water structuring examples, which I guess everyone probably can think of at least some of these. And Pollack gives these examples in cells and gels, is 2 plates of glass, and when they're dry, they're pretty easy to separate. But if there's a layer of water between them, they become more difficult to separate. Or say a difference between wet and dry sand. If you step in dry sand, your foot sinks deeply, leaving an amorphous depression. But if you step in wet sand, your foot barely sinks at all and leaves a fairly crisp foot-shaped impression. So water here is clearly modifying the properties of the sand.
I'll try to include it in the show notes, but there's a really interesting one in The Fourth Face of Water by Pollack, and he talks about water droplets persist on the water surface for some time after they're dropped from like a pipette, and they have a super slow-mo video of it. It's actually pretty incredible, but it's the pipette dropping the little water ball on top of a glass of water and it bouncing off of the surface. And that's one of the examples he used kind of to prove your point, Phil.
Yeah, I think that's a good example. I think an example that Ray Pete has given, which is also very good, it's a very simple example, but I think it still shows evidence of water structuring, is a drop of water moving down an inclined surface, where you'll see a difference between contact angles between, say, the front of the drop, the lower edge, which is moving down, which is more rounded, and then the kind of trail it leaves behind is showing a reluctance of the upper surface to break contact with the surface it's rolling down, and a reluctance of the forward surface to wet the surface that it's moving down. But again, these examples seem kind of so mundane as to be trivial, but I think it's the very fact that they are kind of so mundane that makes them meaningful, because it shows that water structuring isn't something unusual or rare. It's just a very basic property of matter or energy, and it would be very strange if life didn't make use of these properties.
Why do you think these things are just completely ignored just by mainstream science?
I think there might be some ideology ideological concerns involved. Yeah, I think once you start looking at the world like this, it kind of inevitably follows that there maybe isn't such a distinct kind of point where life begins, where dead matter ends and then living matter starts. That we're living in a fundamentally biological and also, I think, biophilic universe. And if it's, if the universe is alive, then it may well be purposeful. This fosters an attitude of reverence for all life. Like as William Blake said, all that lives is holy. Life delights in life. And I think this isn't a model which is particularly compatible with the current ruling ideologies.
I'm going to read a little of this Szent-Györgyi quote. But life originated in the ocean. Water is its matter and matrix, mother, and medium. Life could leave the ocean when it learned to grow skin and take the water with it. We are still water animals, only we have the water inside, not outside. We are a walking aquarium. So the real question is, why is Szent-Györgyi so quotable?
I think it's why is Szent-Györgyi only famous for vitamin C when I think all of his other work is just seems a lot more kind of applicable to life than the current kind of model. I mean, why are his insights into bioenergetics virtually ignored?
His work really is incredible, easy to read, and he's extremely elegant. But back to water.
Okay, yes. So water is composed of hydrogen and oxygen. You've got two hydrogen at one end and then an oxygen at the other. And the oxygen is acting as it withdraws the electrons from the hydrogen. So the hydrogen then become positively charged protons, and this turns the water into a dipole. Because of this, it then has a tendency to form various structures with other water molecules.
It forming structures with other molecules isn't so much controversial. Just how many layers is the controversial thing, correct? Yeah,
I think it was it's basically accepted that there's some limited water structuring occurring in, I think, I'm not sure exactly how many layers they now will concede that structure occurs on, but I think Pollock has shown that the water structuring is extensive.
Yeah, some of his videos are really interesting. I think he says there are millions of them, which is instead of 3 or 4, which is commonly thought, which is pretty interesting.
Yeah, I think the number of layers you're going to get of structured water is going to vary depending on the substance which is structuring them and its size and other factors, including external things like light.
Okay, so let's translate this into kind of in the context of the cell and how. So if there aren't channels and pumps maintaining the ion gradients, how is this kind of new view of water maintaining them?
Okay, so the protein structures the water, and the water then becomes a gel, and it naturally excludes the sodium. The potassium can fit into kind of gaps within the water structure and adsorb to carboxyl groups on the protein, whereas the sodium, which whilst it's a smaller ion than the potassium, has a larger charge density and a larger hydration shell.
And so the proteins, those charge sites, there's competition for them, and because of the smaller shell of the potassium ion, it absorbs to those charge sites on the protein.
And then when the protein is stabilized in that structure, it takes no energy to exclude the sodium. But if something changes, the electric balance of the molecule, or causes it, like something discharges it, it loses ATP, then the protein molecule changes conformation and water is no longer structured in the cell, and so sodium can then enter. In muscle fibers, there's the A band is composed of myosin, and myosin contains most of the carboxyl groups in muscle. Contains more than actin in the I band, and it appears that most of the potassium in muscle fibers is concentrated in the myosin, suggesting that it may well be in association with these carboxyl groups. So I think these findings this has been found using techniques like X-ray microanalysis and other techniques where a section of muscle is prepared for electron micrograph, and then it's vaporized with a laser, and then the vaporized ions are analyzed. Yeah, in experiments where the membrane has been damaged through exposure to solvents, potassium is generally very slow to leak out of the cell, but when it finally does leak, it doesn't kind of trickle out. It comes in this large kind of bolus along with the protein, suggesting that whatever's happened to the cell has just disrupted its structure fairly thoroughly. And Szent-Györgyi has some experiments as well that he was looking at the effects of fluorescent substances, or looking how cytoplasm affected various fluorescent substances, and found that the cytoplasm in its resting state was analogous to the effect that or the way these substances behaved in ice. But when stimulated to contract, the cytoplasm caused the fluorescent material to behave as it would in bulk water. And in this state, when the cytoplasm contracted, there was no fluorescence.
Real quickly, the difference between kind of the structured and bulk water?
Okay, the structured water, there's more structuring, basically, and the bulk water, there's more freedom of the water molecules to move.
Would that just be the lack of proteins and
the lack of the electric charge, I guess.
Exactly. Okay. People will be like, "Oh, this is just a theory," but this associate-induction hypothesis, which we'll get into detail in a second, this helped create the MRI, which is pretty incredible, right?
Yeah, or at least it acted as a major inspiration for the development of MRI. And this is through the work of Ray Damadian, who was familiar with Ling's AI Association-Induction hypothesis, and thought that cancer cells might show evidence of altered water ordering, and thought it might be detectable with magnetic resonance imaging. And Damadian seems to be right, that the water in cancer cells, when you analyze it with NMR, it has an increased relaxation time, suggesting that the water is less structured. And this would, again, go along with these ideas that, or the observation that cancer cells have increased glycolysis, so their energy production is impaired. And this, again, changes the structural properties of the cell.
And that was an observation originally noticed by Otto Warburg?
Yeah, I think so. But yeah, Damadian didn't get any official recognition for his contribution to MRI. And he built, he suggested the creation of NMR machines capable of scanning the body, and built an early one. Although the images it produced, I don't think they were that clear. And the technology was improved by Lauterbur, who received, I think, the Nobel Prize for it. But yeah, Damadian's contribution to the field was ignored. And the fact that MRI works at all might suggest that there's something to Ling's theory. If the images generated are reliant on different relaxation times of hydrogen nuclei or hydrogen, then the relaxation times will vary depending on how tightly water is structured in different tissues.
This is a correspondence from Damadian to Ling, and he says, on the morning of July 3rd, 4:45 a.m., we achieved with great jubilation the world's first MRI image of the live human body. The achievement originated in the modern concepts of saltwater biophysics, on which you are the grand pioneer with your classic treatise, the Associate-Induction hypothesis. So this is a pretty good segue into the specifics of Ling's Associate-Induction hypothesis. Phil, do you want to go over the oscillation in the living states, the active living state and the resting living state, and how ATP is integral to that process?
Okay, so yeah, according to Ling, ATP is what he calls the queen of cardinal adsorbents. And the cardinal adsorbent is a molecule that, through its association with a protein, is able to induce electrical changes in the protein, changing both the conformation of the protein and the way it interacts with water. So for proteins to control the water in the cell and keep it orderly and excluding sodium, they have to be extended. And to do this, ATP has to be present to withdraw electrons from the protein.
Ling has an interesting drawing of this in his book, but basically it's like a squiggly line, and the squiggly line represents the protein. And that's when ATP is not sitting on the cardinal site of the protein. And then the next depiction is a straight line, and that's when the ATP is sitting on the cardinal site of the protein. And it's oscillating between those two states.
Yeah, once ATP is restored the cardinal site on the protein can then re-extend and order water again, and you can return to the relaxed, high-energy resting state. But if something interferes with ATP production, then that kind of active living state, the active state, basically becomes kind of a pathological state of, say, for example, muscle cramp, or nerve paresthesias, and can contribute to things like insomnia. I think an interesting observation in this is Szent-Györgyi noticed that muscles in a state of rigor mortis, when they are placed in a solution of ATP, they'd soften.
And Pete often in interviews, he says kind of the first thing when a cell is damaged, it takes up water. So this would explain that kind of phenomena, the active living state not being able to really maintain ion gradients and taking in the sodium and calcium outside and losing the intracellular potassium and magnesium.
Yes, exactly. Yeah, if energy production is interfered with, then the cell will swell and take up water, and that tends to become a self-reinforcing situation.
I mean, this changes how the cell is energized, right? This
is like a good point. I've never really been able to figure out how ATP works in the standard model. I mean, there's this talk of a high energy phosphate bond, but I don't understand how it produces work, how it contributes to function. How does ATP in the standard model work? Within Ling's model, it seemed the whole thing seems to make sense and seems fairly consistent with kind of living experience, and and would explain observations like Achilles tendon reflex test for hypothyroid. The muscle, calf muscle in a hypothyroid condition is going to be slower to relax fully because energy production is impaired. So ATP isn't going to be able to complex with protein and order water and allow the tissue to relax.
I think Pollack says ATP binding is so tight that twice as much energy is released compared to hydrolysis of ATP being broken down into ADP and inorganic phosphate. Did Szent-Györgyi say the living state is electronically desaturated state?
Yeah. I think this kind of, again, fits with Szent-Györgyi's work. Like, if ATP is acting as an electron withdrawing cardinal adsorbent, then it's desaturating the protein. And that's the living state, is the electronically desaturated state, which is relaxed, it's high energy, and that gradual disorder of this living state is like the basic feature of aging.
What do you think about that? Szent-Györgyi noted the life expectancy of an individual by pinching the skin on the back of their hands.
Yeah, I think again that observation is consistent with this. It's showing how efficient the organism is at producing energy, and again, restoring itself to the living state.
So, Phil, what do you think some of the practical implications of this are for health and disease? Like, how do you maintain ATP, and what happens when you can't generate energy efficiently?
Okay, we've already kind of touched on some of this, like with mentioning cancer and the increased relaxation times of water molecules, and noting that in aging, there's a decreased relaxation time in the skin, and this information is obviously relevant to health. It seems to be a much more kind of realistic model than the kind of lipid membrane model, which seems innately mechanical. And I think this kind of mechanistic view of life kind of leads to a lot of mistaken ideas. Like, oh, look, there's cholesterol in fatty plaques and atherosclerosis. Cholesterol must cause heart disease, and we better lower cholesterol at all costs. And soft tissues calcify. The observation that soft tissues calcify in multiple diseases. So, eat a low calcium diet. Whereas, in Ling's model, the observation is, well, the soft tissues are calcifying because energy charge is disappeared, and if you can restore energy production, then that calcification problem should be prevented or improved.
Yeah, I think Pete has mentioned once or twice that calcification begins in the cell, and then using Gilbert Ling's Associate-Induction hypothesis, it really begins to make sense how you can see the intracellular accumulation of calcium when the cell is stressed or injured or dying or whatever.
It's remarkably consistent with many observations. Inflammation in general seems to be like a state of the cell's energy production is impaired. It's fatigued. And they take up water. This also explains the observations like the permeability issues in things like leaky gut. The barrier function of the intestine is decreased. Maybe in part because of increased exposure to things like endotoxin, which impair energy production, and the cell then becomes unable to return to the relaxed resting state where it's ordering water. And if it can't do that, then you end up with this highly permeable leaky gut.
Do you want to briefly go over redox state? I know it's a loaded question.
Okay.
But there's some evidence.
Yeah. There is some evidence that cancer has a characteristic redox state. And, this again is the idea that it's more reduced than non-cancer cells. So this can be seen probably most obviously in the ratio of NADH to NAD, which I think there's a significant difference between cancer and non-cancer cells, where in cancer cells, NADH it's massively in favor of the NADH rather than the NAD. And again, this is suggesting a kind of a discharged state.
That's so there's not enough agents withdrawing electrons, and that's creating oxidative stress?
Yeah, and the fact that there's not enough agents withdrawing electrons and the cell is becoming disordered is then going to further interfere with energy production and prevent the issue from resolving. But if you can restore the kind of the redox balance and the cell's charge with agents that withdraw electrons such as ATP, carbon dioxide, or progesterone, then the cell, you might be able to return the cell to its highly energized resting state. And this seems probably like a solution that is so simple that it's too good to be true. But there is in vitro evidence of redifferentiation of cancer cells into non-cancer cells through using agents such as progesterone, butyric acid, and DMSO. The fact that you can trigger the redifferentiation of cancer cells into non-cancer cells suggests that there is an entirely other approach to treating cancer. And this just seems to be almost entirely ignored in research on cancer. This would be a a therapeutic that would surely be basically benign. No toxic chemotherapy and radiotherapy, but no, it's ignored. I mean, and cancer is just, one problem. There's, other issues where you can see similar processes resulting. And again, a bioenergetic model like Ling's seems to be consistent with what's seen. So in the heart, stress and low energy result in uptake of calcium and a state of continuous partial contraction. Full relaxation and full contraction are both impaired, and the heart will beat rapidly but weakly, and eventually this state results in fibrosis, and the blood vessels in this state can't relax, and you get hypertension and heart failure. But yeah, I've mentioned nerve problems like paresthesias and insomnia, and this is all because the cytoplasm is unable to shift back to the resting state. Basically, I think Ling's model is highly applicable to multiple conditions, possibly all conditions.
Okay, so not only like the Achilles reflex test, but also like you mentioned a couple times, like cramps, and I don't know the actual name of this, but like when you do sudden kicks. Phil, do you know the medical term for that? If you're like laying down before you go to bed and you like kick real hard. Pete mentioned it in an interview. I forget what it's called. But he thought all of those were kind of electro disturbances in the cell, at the cell level.
Yeah, I think so, either in the muscles or the nervous system itself, yeah. And I think, like the use of magnesium in conditions like cramping would explain why it's working, because it complexes with ATP and helps to kind of stabilize the resting state. And
there are a few people in the health world that have anything negative to say about magnesium, which is interesting.
Yeah, it's generally a fairly safe substance to talk about without too much controversy.
Yeah.
And also I think this model kind of explains Ray Peat's kind of stance on PUFAs, which he's kind of famous for, or infamous for.
So these are the unsaturated fats, like vegetable and seed oils.
Yeah. Again, Ling proposes that fats, whilst they're not serving any function in any lipid bilayers, as the lipid bilayer doesn't exist, but in Ling's model he does allow for a membrane, but it's not a lipid bilayer membrane. It's composed of protein and structured water, but it's not substantially different to the main kind of meat of the cell.
I mean, I've heard people, they'll be like, you need these essential fatty acids because they make up the membranes of your cells.
Yeah, you'll see all kinds of crazy stuff like, if you don't have, flexible membranes, you'll become diabetic because saturated fats stop sugar from getting in the cell. And, I've seen, health books with this stuff written in. There's no references for this, oddly enough. So yeah, basically Ling proposes that saturated fats are capable, through their hydrophobic interactions, of stabilizing cellular proteins. And Ray Peat has pointed out that PUFA, because of their unsaturations, are relatively more hydrophilic, and they're going to modify the way water and proteins interact, and also cause things like cross-linking of proteins. And it's these kind of possibilities which might explain observations like those of Nanji and others who found that saturated fats like butter, coconut oil, and were capable of preventing and even reversing liver cirrhosis, even whilst alcohol administration was ongoing, whilst the PUFA promotes cirrhosis. And also, saturated fat intake is, in some studies, inversely correlated with tumor invasiveness. Palmitic acid, a saturated fat, is an important lung surfactant, and the PUFA cause edema water uptake in the lungs. Yeah, there's some evidence also that the saturated fats are capable of inhibiting glycolysis in the cell, suggesting they're stabilizing the cytoplasm. Yeah, the highly unsaturated PUFA, DHA, and arachidonic acid, they can be seen. You can see evidence that they're acting to disturb structure because they increase cellular calcium level, and this will decrease the cellular sensitivity, possibly suggesting that they've subtly shifted the cell towards a more excited state that it can again entirely relax, and this will decrease overall cellular sensitivity.
And the PUFA modifying the way the water and proteins interact, that would just mean that it was inhibiting the production, the charge of the protein and the production of energy?
Yeah. It's going. It's just going to disturb cell function on a fairly fundamental level, and make cells less capable of returning to the resting living state. This kind of this model of looking at the cell as protoplasm rather than some kind of membrane-bound sack of water, wherein you've got I mentioned earlier the problems with explaining the functions of things like estrogen and progesterone through membrane-bound receptors. In experiments with rabbit hearts, Szent-Györgyi showed that estrogen decreased the heart's readiness to work and that progesterone increased its readiness to work. And Szent-Györgyi thought it did this, or these substances, in the case of progesterone, it did it by building structure, and in the case of estrogen, it did this by disturbing structure. And he pointed out that for a given drug or stimulus, cells have a characteristic response becoming either more activated or more inhibited.
But Phil, if the heart works less, that means it's better off, right?
I don't think so, but not necessarily, as decreased capacity to work is probably going to be associated with increased calcium uptake and decreased energy production. Estrogen does appear to be clearly excitatory, and I don't see how you can explain this through receptors. I mean, receptors kind of then knock some kind of little push-button receptor gets activated, and it knocks on a protein, which knocks on another protein, and eventually switches on some gene in the nucleus and triggers some other set of odd clockwork responses. Yeah, again, there's this strange idea that you need, people talk about ideas of, hormone balance or a neurotransmitter balance, and you've got to have the right neurotransmitter balance, because if you don't have a certain amount of the right excitatory substance pressing the right number of receptor buttons, you can't maintain balanced homeostasis. And I think this idea of balance completely ignores that many of these substances are intimately involved in inflammation, not only triggering it, but released during inflammation and injury. None of this is surprising when you realize how kind of deeply independent or interdependent energy and structure are. Injury disrupts structure, disrupting the flow of energy, or and triggering a energetic discharge. And then this allows for the release of serotonin, say, which otherwise, in a state of health and energy flow, would have remained bound and inactive. And all of these transitions, like say the release of say serotonin, histamine from a mast cell, this is mediated through a phase transition, conformation change in the protein and water structure of the mast cell. Will, when its structure is disrupted, it will then release the serotonin and histamine. And these substances are clearly inflammatory and are clearly released by inflammation. I don't think it's possible for serotonin to get too low. I mean, if there's inflammation, it's going to be released. And it seems that what serotonin does is inflammation. Somehow people have got the idea that it's the happy hormone. I don't think you can really understand what is going on with these substances unless you're thinking in a coherent way which accounts for the basic properties of matter and energy and incorporates like water structure into this model. Because if you're thinking about like the organism as some kind of strange machine, I think it's very easy to get confused and to think that these molecules are just acting as little signal substances and they're just transmitting some sort of abstract information rather than acting primarily energetically. I think it's important to develop a realistic view of life. I think if you think life is mechanical, it's probably going to distort the way you interact with the world. And yeah, it's definitely distorting medicine. Certainly, obviously both of us are clearly biased and are fully convinced that bioenergetic models of people like Ray Peat and Gilbert Ling are basically fairly accurate. I mean, there's probably room for fine-tuning and some refinement, but I think the basic models that they present are close to the truth. I think this is also relevant. There's a quote from Ray that's probably relevant here. He says on his website, "I think only a new perspective on the nature of living matter will make it possible to properly take advantage of the multitude of practical and therapeutic effects of the various life-supporting substances, pregnenolone, progesterone, thyroid hormone, and coconut oil in particular."
This seems like a good place to wrap things up. Phil, I want to sincerely thank you for joining me today. I can't believe how much information we covered. If you want to find more of Phil's work, you can go to pranarupa.wordpress.com, pranupa.wordpress.com, and you can find all my work at dannyroddy.com. See you guys later. Bye.